Tonight, we're going to slow down and take a quiet journey into one of the most extraordinary things in the universe, the code hidden inside every living cell. So, wherever you are right now, maybe lying in bed, maybe studying late, maybe listening on a quiet train home, I'm really glad you're here. Settle in, let your mind drift gently, and together, let's explore how a tiny strand of DNA became you. Close your eyes for a moment and imagine something almost impossibly small. Smaller than a grain of sand, smaller than a speck of dust floating through the air. Inside your body right now, there are trillions of tiny living cells, quietly working, quietly communicating, quietly keeping you alive. Every breath you take, every thought you think, every beat of your heart depends on these microscopic structures. And hidden inside almost every one of those cells is something extraordinary. A long molecular instruction system known as DNA. DNA is not alive in the way cells are alive. It does not think. It does not make decisions. But it stores information. Vast amounts of information. Information about how to build proteins, how to organize cells, how to grow tissues, how to maintain the body over time. In a very real sense, DNA is a biological archive, a molecular library carried inside your cells from the moment life began. Right now, inside your skin cells, your blood cells, your muscle cells, and your brain cells, nearly the same complete set of DNA instructions is present. Even though those cells perform very different jobs. A skin cell behaves nothing like a neuron. A liver cell behaves nothing like a muscle fiber. And yet, deep inside, they contain almost the exact same genetic information. This idea surprised scientists for a very long time, because the human body is incredibly diverse. Your eyes contain specialized light-sensitive cells. Your immune system contains defensive cells that patrol your bloodstream. Your bones contain cells that constantly rebuild microscopic damage. And still, the same DNA blueprint exists inside nearly all of them. The human body begins from a single fertilized cell. One cell, tiny and invisible to the naked eye. That single cell divides into 2, then 4, then 8, then 16. Over and over again. Eventually forming trillions of cells organized into tissues and organs. The brain, the lungs, the heart, the skin, all emerging from that first tiny beginning. And through all of those divisions, DNA is copied again and again, carefully replicated, cell after cell, generation after generation. Your DNA today is connected to a chain of life stretching back billions of years. Through parents and grandparents, through ancient humans, through mammals, through early life in the oceans of Earth. DNA is remarkably ancient, not just in humans, but across almost all known life. Trees use DNA. Birds use DNA. Fish use DNA. Bacteria use DNA. The exact sequences differ, but the underlying system is shared. which means that deep beneath the enormous diversity of life, there is a common molecular language. Scientists did not always know this. For most of human history, nobody understood how traits were inherited. People noticed patterns. Children often resembled their parents. Certain illnesses seemed to run in families. Farmers observed that selective breeding could shape plants and animals over generations. But the actual mechanism remained hidden. In the 19th century, an Austrian monk named Gregor Mendel began studying pea plants. He carefully tracked traits across generations: flower color, seed shape, plant height. His experiments revealed that inheritance followed patterns. Traits were passed down through discrete units. Today, we call those units genes. Decades later, scientists discovered chromosomes inside cells. And eventually, they identified DNA as the molecule carrying genetic information. In the 1950s, researchers uncovered the famous double helix structure of DNA. Two intertwined strands forming a twisted ladder shape. Stable enough to store information, flexible enough to replicate, compact enough to fit inside microscopic cells. The amount of DNA packed into your body is difficult to imagine. If you stretched the DNA from a single human cell into a straight line, it would measure roughly 2 meters long. And yet it fits inside a nucleus far too small to see without a microscope. Your body contains trillions of cells, which means the combined length of all your DNA would extend incredibly far, far beyond everyday scales. All compressed into living structures smaller than dust particles. DNA works chemically. It is made from smaller units called nucleotides. Each nucleotide contains one of 4 chemical bases: adenine, thymine, cytosine, and guanine, often shortened to A, T, C, and G. These 4 molecular letters form sequences, and those sequences contain instructions, very much like letters forming words, and words forming sentences. Except, instead of creating language, DNA sequences help create proteins and biological structures. What makes DNA remarkable is not the complexity of its alphabet. The alphabet is actually very small. Only 4 chemical letters. The complexity comes from the arrangement, the order, the patterns. Just as 4 musical notes repeated in different ways could create countless melodies, 4 DNA bases arranged in long sequences can encode immense biological variety. Human DNA contains around 3 billion base pairs. 3 billion molecular steps written into nearly every cell of your body. Most of those sequences are shared among all humans. In fact, any 2 humans are genetically extremely similar. But small differences in DNA contribute to variation. Height differences, eye color differences, metabolic differences, even differences in disease risk. DNA is constantly protected by the cell because it is essential information. Cells contain repair systems that monitor DNA for damage. Sunlight can damage DNA. Certain chemicals can damage DNA. Normal cellular processes can accidentally introduce copying errors. so cells continuously inspect and repair their genetic material. Most of the time, these repair systems work remarkably well. Your body is maintaining and correcting DNA right now, quietly, without your awareness. Thousands upon thousands of microscopic chemical events happening every second. And yet DNA is not a rigid script controlling every detail perfectly. Biology is more flexible than that. Genes interact with environment. Nutrition matters. Sleep matters. Stress matters. Development matters. DNA provides instructions, but living systems constantly respond to conditions around them. This balance between stability and flexibility is one reason life can adapt. Organisms survive changing environments. Species evolve over long periods of time. Cells specialize into many forms, all built upon the same fundamental molecular language. Perhaps one of the most fascinating ideas in biology is that your body is reading molecular instructions every moment of your life. Right now, proteins are being assembled. Cells are communicating. Old molecules are being recycled. Damaged cells are being replaced. The body is active even during sleep, especially during sleep. While you rest, DNA continues quietly guiding cellular processes. Skin repairs itself. Immune cells remain alert. Memories are reorganized in the brain. Life continues in silence beneath conscious awareness. It is easy to think of ourselves as singular beings. One person, one mind, one body. But biologically, we are vast, living systems made from unimaginable numbers of microscopic interactions. And DNA sits quietly near the center of that system. Not as magic, not as destiny written perfectly in advance, but as stored biological information shaped by billions of years of evolution. Every living thing on Earth today carries some version of this ancient molecule, passed forward continuously across deep time, from cell to cell, from parent to child, across generations too vast to truly picture. And before we can understand how genes shape the body, we first need to look more closely at the structure of DNA itself. If we could gently travel inward, past the surface of the skin, past layers of tissue, down into a single microscopic cell, we would eventually arrive at a small structure near its center, the nucleus. And inside that nucleus, packed carefully into an incredibly tiny space, is DNA. DNA is often described as a twisted ladder. Scientists call this shape a double helix. Two long strands wrapped around each other in a slow spiral. Elegant, stable, efficient. The shape matters greatly, because DNA must do two things at once. It must store information safely, and it must also copy itself accurately when cells divide. The double helix structure helps it accomplish both. Imagine a ladder that has been gently twisted along its length. The sides of the ladder form the outer backbone of DNA. These backbones are made from repeating sugar and phosphate molecules linked together chemically. strong enough to protect the information inside. Between the two sides of the ladder are the rungs, and these rungs contain the famous chemical bases: adenine, thymine, cytosine, and guanine, A, T, C, and G. These bases pair together in very specific ways. Adenine pairs with thymine. Cytosine pairs with guanine. Always. A with T, C with G. This pairing rule is one of the most important features of DNA because it allows information to be copied reliably. One strand can serve as a template for rebuilding the other. When a cell prepares to divide, the two strands separate slightly, like a zipper slowly opening. Each exposed strand then guides the formation of a new matching strand. A pairs with T. C pairs with G. Over and over again. This process is called DNA replication. It happens continuously throughout your life. Skin cells divide. Blood cells divide. Cells lining the digestive system divide. And before each division, DNA must be copied with extraordinary precision. The accuracy is remarkable. Errors do happen occasionally, but cells contain proofreading and repair systems that reduce mistakes dramatically. Most DNA replication occurs with astonishing reliability. The information stored in DNA depends entirely on sequence. The order of the bases matters. A single change in sequence can sometimes alter how a gene functions. Much like changing a single letter in a sentence can change its meaning. But unlike human language, DNA does not use words or grammar. It uses chemistry, molecular interactions, physical structures, binding patterns. The cell interprets DNA through biochemical processes. DNA itself is surprisingly simple at the chemical level. The complexity of life emerges from enormous combinations built from simple repeating parts. This pattern appears often in nature. Simple atoms forming complex molecules. Simple notes forming symphonies. Simple bases forming genomes. The human genome contains roughly 3 billion base pairs. That means around 3 billion steps in the molecular sequence. If written out as letters in a book, it would fill enormous volumes. And yet nearly every cell in your body carries this information, folded tightly and organized carefully inside chromosomes. DNA packaging is another remarkable feature of biology. Because DNA is very long, far too long to simply float freely inside the nucleus. So cells wrap DNA around proteins called histones, almost like thread wound around tiny spools. These wrapped structures coil and fold repeatedly, becoming more compact, more organized, more manageable, eventually forming chromosomes. Humans usually have 46 chromosomes in most body cells, organized into 23 pairs. One set inherited from the mother, one set inherited from the father. Inside these chromosomes are genes, specific stretches of DNA containing instructions for building functional products, usually proteins. But genes make up only a portion of total DNA. Much of the genome helps regulate activity or plays structural and regulatory roles scientists are still studying. Early researchers once assumed most DNA directly coded for proteins. But biology turned out to be more complicated. Some DNA sequences regulate when genes activate. Some help organize chromosomes. Some produce functional RNA molecules rather than proteins. And some regions remain only partially understood. The more scientists study genetics, the more they discover that DNA functions as part of an interconnected system, not isolated instructions acting independently, but networks interacting constantly inside living cells. Even the shape of DNA matters beyond information storage. Certain proteins recognize particular DNA sequences by physical structure and chemical attraction. Molecules attach temporarily to DNA. Others detach. Sections become more accessible or more tightly packed. The genome is dynamic, active, continuously managed by the cell. DNA is also directional. The two strands run in opposite orientations. This arrangement is important for replication and for reading genetic information correctly. Many cellular enzymes operate only in one direction along a strand. Specialized proteins constantly move along DNA, performing different tasks. Some copy it. Some repair it. Some help read genes. Some help package it. The nucleus is never truly inactive. Even now, while you listen quietly, countless molecular interactions are unfolding inside your cells. Invisible, silent, continuous. DNA strands opening and closing, proteins attaching and releasing, information being copied and interpreted. Scientists uncovered the double helix structure in the 20th century using multiple lines of evidence. X-ray diffraction images played a major role. These images revealed repeating patterns suggesting a helical structure. Researchers including Rosalind Franklin, James Watson, and Francis Crick contributed to understanding DNA's structure. Their work transformed biology permanently. Once scientists understood the structure of DNA, many mysteries of inheritance became clearer. The pairing rules explained replication. Sequences explained stored information. Genes could now be understood physically rather than abstractly. And, over time, entirely new scientific fields emerged. Molecular genetics, genomics, biotechnology, genetic medicine, all built upon understanding this molecular structure. Yet, despite everything modern science has discovered, DNA still retains a quiet elegance. 4 chemical bases, repeating patterns, complementary strands, simple physical rules. From these ingredients emerges the enormous complexity of life. Every animal, every plant, every human being, every microscopic bacterium, all using variations of the same molecular system. The DNA inside your cells right now is not separate from the history of life on Earth. It is part of that history, continuously copied across ancient generations, stretching back through deep evolutionary time. And hidden within those long DNA strands are smaller, functional units that carry specific biological instructions. Inside the long strands of DNA, there are smaller regions carrying specific instructions. These regions are called genes. A gene is not a separate object floating inside the cell. It is a sequence within DNA itself. A particular arrangement of bases. A specific molecular pattern stored along the strand. Some genes are very short. Others are much longer. Each contains information that cells can use to help build functional molecules, most often proteins. You can think of DNA as an enormous library, and genes as individual instruction pages within that library. Not every page is read at once. Not every instruction is needed in every cell. But together, they form the genetic blueprint of the organism. Humans are estimated to have around 20,000 protein-coding genes, far fewer than scientists once expected. Early researchers assumed human complexity would require enormous numbers of genes. But biology turned out to rely heavily on regulation, interaction, and reuse of genetic instructions. A single gene can sometimes contribute to multiple functions, and many traits arise from interactions among numerous genes working together. Height is one example. There is no single height gene. Instead, many genes contribute small effects related to growth, hormones, bone development, metabolism, and cellular signaling. Environment also matters. Nutrition during development matters. Health matters. Most biological traits emerge from combinations of influences rather than simple one-gene control. Genes are organized into chromosomes. Chromosomes are long package structures made from DNA and proteins. They help organize and protect genetic material inside the nucleus. Humans typically have 46 chromosomes in most body cells, arranged into 23 pairs. One chromosome of each pair comes from the mother. The other comes from the father. This is one reason children resemble both parents. They inherit combinations of genetic variants from each side of the family. Sometimes certain traits resemble one parent more strongly. Sometimes traits blend more subtly across generations. The first 22 chromosome pairs are called autosomes. The remaining pair are the sex chromosomes, usually XX in females, usually XY in males. Though biological variation can occur naturally in sex chromosome patterns as well. Each chromosome contains many genes. Some chromosomes carry thousands. Others carry fewer. Different chromosomes contain instructions related to many different biological processes. Certain genes influence eye development. Others help regulate the immune system. Others participate in brain function, metabolism, or cellular repair. Many genes are active across multiple tissues simultaneously. Yet, even though nearly every cell contains the same chromosomes, cells behave very differently from one another. A neuron does not behave like a skin cell. A muscle cell does not behave like a liver cell. This happens because cells activate different sets of genes. Some genes are turned on. Others remain inactive. The underlying DNA stays mostly the same, but gene activity changes from cell type to cell type. This selective gene activity is one of the foundations of multicellular life. It allows specialization, division of labor, complex tissues, organized organs, entire biological systems working together. During early development, this process begins very quickly. A fertilized egg divides repeatedly. At first, the cells are relatively unspecialized. But gradually, chemical signals and gene regulation guide cells toward different roles. Some become nerve cells. Some become muscle tissue. Some become blood cells. Some help form organs, all emerging from the same original genome. This process is called differentiation. Cells gradually becoming more specialized over time. One of the most remarkable processes in biology. The instructions encoded in genes are not interpreted automatically. Cells must first read those instructions. Specialized proteins bind to particular DNA regions and help regulate activity. Some genes become accessible. Others remain tightly packed and inactive. In many ways, the genome functions less like a static blueprint and more like a dynamic reference system. Different sections are used at different times under different conditions. Development changes gene activity. Environment changes gene activity. Aging changes gene activity. Even throughout a single day, patterns of gene expression can shift. Some genes follow circadian rhythms linked to sleep and light cycles. Others respond to stress hormones, nutrition, temperature, or physical activity. Your cells are constantly adjusting, quietly regulating internal processes in response to changing conditions. Scientists use the term genome to describe the complete set of genetic material in an organism. The human genome includes all human DNA sequences together. Protein coding genes, regulatory regions, structural regions, repetitive sequences. Everything contained within the full genetic archive. Mapping the human genome became one of the great scientific efforts of the late 20th century. The project involved researchers from multiple countries working together to sequence human DNA. This became known as the Human Genome Project. The goal was ambitious. Identify the sequence of human DNA bases across the genome. Build reference maps. Improve understanding of genes and disease. The project officially began in 1990. And by the early 2000s, scientists had produced a major draft of the human genome sequence. It transformed biological research. Suddenly, researchers could study genes more systematically, compare genomes between individuals and species, investigate inherited disorders more deeply, explore evolutionary relationships with new precision. Yet the genome did not provide simple answers to every question about biology. Scientists quickly realized that understanding genes alone was not enough. The timing of gene activity mattered greatly. Interactions mattered. Cellular context mattered. Biology remained deeply interconnected and complex. Still, the genome offered an extraordinary new window into life. A record of inheritance stretching across generations. a molecular history written inside cells. Humans share most of their DNA with one another. And, surprisingly, humans also share large portions of genes with many other organisms. We share genes with mice, with fish, even with plants and bacteria at more distant levels. This shared biology reflects common ancestry across evolution. Life diversified over immense spans of time, but many fundamental molecular systems remained conserved. Certain genes involved in cellular energy production exist across enormous branches of life. Some mechanisms controlling DNA replication are shared broadly among organisms. The core chemistry of life is ancient. Even the genetic code itself is nearly universal. Across most organisms on Earth, similar DNA sequences correspond to similar amino acids during protein construction. This consistency helps reveal how deeply connected living systems truly are. And although genes contain instructions, they are only the beginning of the story. Because instructions alone do not build tissues directly, cells must first decide which genes to use and when to use them. Every human life begins quietly. Not with organs, not with thoughts, not with movement, but with a single cell. A fertilized egg cell formed when genetic material from two parents combines together. Inside that tiny cell is a complete human genome. 46 chromosomes carrying inherited DNA instructions. At this stage, the cell is extremely small, invisible without magnification. And yet within it exists the molecular potential for an entire human body. The first major process that begins is cell division. The fertilized cell divides into 2 cells, then 4, then 8, then 16, again and again. This process is called mitosis. During mitosis, DNA is copied carefully so that each new cell receives a nearly complete set of genetic instructions. The genome must be replicated with extraordinary accuracy. Very early on, these cells are relatively unspecialized. They have not yet become skin or muscle or nerve tissue. Scientists often refer to many early developmental cells as stem cells because they still retain the ability to become different cell types. As division continues, groups of cells begin organizing into patterns. Chemical signals move between neighboring cells. Some genes become active. Others become inactive. Gradually, differences begin to emerge. This process is known as differentiation. Cells becoming specialized over time, taking on distinct structures and functions. Some cells begin moving toward becoming neurons, others toward muscle tissue, others toward blood cells, others toward organs and connective tissues. One of the most remarkable facts in biology is that these different cells still contain nearly the same DNA. A neuron and a skin cell usually carry the same genome. The difference lies mainly in which genes are active. Cells do not read the entire genome all at once. Instead, they selectively use certain instructions depending on their role. A developing eye cell activates different genes than a developing liver cell. Different proteins are produced. Different structures emerge. This selective gene activity is coordinated through networks of molecular signals. Cells communicate constantly during development. They release signaling molecules, detect chemical gradients, respond to nearby tissues. Development is highly interactive. No single cell understands the entire body plan. Instead, complex structures emerge gradually from countless local interactions happening simultaneously. Early in development, one major event is the formation of three primary tissue layers. These are called the ectoderm, mesoderm, and endoderm. The ectoderm eventually contributes to structures like the nervous system and outer skin layers. The mesoderm contributes to muscles, bones, blood, and connective tissues. The endoderm helps form internal organs, such as parts of the digestive and respiratory systems. From these early layers, increasingly complex structures begin to emerge. The nervous system starts forming very early. Primitive heart tissue begins developing. Small folds and structures gradually organize into organs. None of this happens instantly. Development unfolds step by step over time. Genes activate in sequences. Cells respond to neighboring cells. Structures influence one another during growth. Timing is extremely important. Certain genes must activate at precise stages. Some signals appear only briefly during development. Small disruptions in timing can sometimes alter developmental outcomes significantly. Scientists study these processes through developmental biology, a field focused on how complex organisms emerge from simple beginnings. It combines genetics, molecular biology, anatomy, and cellular signaling. One important discovery is that many developmental genes are deeply ancient. Certain gene families involved in body organization appear across multiple animal species. This suggests that evolution reused and modified existing developmental systems over immense periods of time. For example, some genes helping organize body structures in humans have related versions in other animals as well. Not identical outcomes, but related molecular tools adapted differently across evolution. Development also depends heavily on physical organization. Cells are not simply floating randomly. They attach to surrounding structures, move along chemical pathways, fold into layers and shapes. Mechanical forces help shape the growing body too. As tissues expand, blood vessels begin forming to deliver oxygen and nutrients. The circulatory system becomes increasingly important as the organism grows larger and more complex. Eventually, organs become more specialized. The heart develops chambers. The brain forms distinct regions. Limbs emerge. Sensory systems organize gradually. Throughout all of this, cells continue dividing, continue communicating, continue activating and silencing genes in carefully regulated patterns. Even after birth, development continues for many years. The brain continues forming connections. Bones grow and remodel. Hormonal systems mature. Immune systems learn from exposure to the environment. Human development is not a single event. It is an extended biological process unfolding across decades. Some cells in the body live only briefly. Others survive for years. Certain tissues constantly renew themselves. Cells lining the intestine are replaced frequently. Skin cells are continually shed and renewed. Blood cells are produced continuously in bone marrow. Meanwhile, many neurons in the brain can persist for extremely long periods. The body is always changing at the cellular level, even while maintaining overall continuity. Development is guided by both genetic instructions and environmental conditions. Nutrition matters greatly during development. Oxygen supply matters. Hormones matter. Exposure to harmful chemicals can matter. Genes provide biological possibilities, but environment helps shape outcomes. Scientists sometimes describe development as a conversation between genes and surroundings. Neither acts entirely alone. Cells constantly respond to internal and external signals throughout life. Even identical twins, who begin with extremely similar DNA, can gradually develop differences over time due to environment, experience, and gene regulation. Another remarkable aspect of development is self-organization. Cells can collectively form complex structures without any central controller directing every movement individually. Local interactions produce large-scale organization. This principle appears throughout biology. Ant colonies organize collectively. Neural networks organize collectively. Developing tissues organize collectively. Complexity emerges from many smaller interactions occurring together. By adulthood, the human body contains trillions of cells. Different shapes, different functions, different lifespans, all coordinated into one living system. And all of it traces back to that first microscopic cell-carrying DNA instructions inherited across generations. Yet possessing genes alone is not enough to explain how cells behave differently. The crucial question becomes how cells decide which genes to use and which genes to remain silent. Inside nearly every cell of your body, the same DNA is present. The same chromosomes, the same genome. And yet, the cells themselves can be completely different. A neuron can send electrical signals across long distances. A muscle cell can contract and generate force. A white blood cell can recognize signs of infection. A skin cell can form protective outer layers. Same DNA, different behavior. The reason lies in gene expression. Gene expression refers to the process through which cells use genetic information. Some genes are active, others are inactive. Some are used constantly, others only under specific conditions. The genome is not read all at once like a book from beginning to end. Instead, cells selectively access certain instructions when needed. You can think of the genome as a vast library. Different cells open different pages. A liver cell reads one collection of instructions. A neuron reads another. An immune cell reads another. This selective reading allows specialization. Without gene regulation, multicellular life, as we know it, would not exist. Gene expression begins when a section of DNA becomes accessible to cellular machinery. DNA is tightly packaged inside the nucleus. Not every region is equally exposed at all times. Some sections remain tightly packed and difficult to access. Others loosen and become more available. Specialized proteins help regulate this process. Certain proteins bind to DNA near genes and influence whether those genes become active. Some proteins increase activity, others reduce it. These regulatory proteins are often called transcription factors. They help control when and where genes are expressed. Cells respond constantly to signals, hormones, nutrient levels, stress signals, chemical messages from neighboring cells. All of these can influence gene expression patterns. Even light exposure can affect gene activity. Your circadian rhythm, the internal timing system linked to day and night, depends partly on changing patterns of gene expression. Certain genes become more active during waking hours. Others increase activity during sleep. The body constantly adjusts its internal chemistry according to time and environment. This regulation is highly coordinated. Genes rarely function in isolation. Networks of genes interact together. One gene may influence another. Proteins produced from one pathway may regulate different pathways elsewhere in the cell. Biology operates through interconnected systems, not through single instructions acting alone. During development, gene expression changes dramatically over time. Early embryonic cells express genes associated with rapid growth and flexibility. As tissues specialize, different expression patterns emerge. Neurons activate genes involved in communication and electrical signaling. Muscle cells activate genes involved in contraction. The underlying DNA remains mostly the same. What changes is how the cell uses the information. Some genes are expressed in almost every cell continuously. These are often called housekeeping genes. They help maintain essential cellular functions like energy production and basic metabolism. Other genes are much more specialized. They may activate only in certain tissues or only during certain stages of life. Gene expression can also change in response to experience. Exercise can alter expression patterns in muscle cells. Learning can influence gene activity in neurons. Immune responses involve large shifts in expression across many genes. Your cells are not static. They continuously adapt to changing conditions. One important layer of regulation involves chemical modifications associated with DNA and its packaging proteins. These modifications can influence whether genes become easier or harder to access. This area of biology is called epigenetics. The word literally means above genetics. It refers to mechanisms that affect gene activity without changing the DNA sequence itself. For example, small chemical groups called methyl groups can attach to DNA in certain regions. This can reduce activity of nearby genes. Histone proteins around which DNA is wrapped can also be chemically modified. Some modifications loosen DNA packaging, others tighten it. These structural changes influence accessibility. Epigenetic regulation is important during development. It helps stabilize cell identity over time. Once a neuron becomes specialized, many expression patterns are maintained consistently, so the cell continues functioning properly. At the same time, some flexibility remains. Cells can still respond dynamically to new conditions. Environmental factors can influence epigenetic patterns. Nutrition, chronic stress, sleep quality, exposure to toxins. All may affect aspects of gene regulation to varying degrees. Scientists continue studying how strong these effects are and how long they persist. The field remains active and complex. Importantly, changes in gene expression do not usually rewrite the DNA sequence itself. The underlying genetic code generally remains stable. What changes is how the cell interprets and uses that code. Some genes must be regulated very carefully because small imbalances can create problems. Too much activity or too little activity may disrupt normal cellular function. Cancer, for example, often involves abnormal regulation of genes controlling cell growth and division. Mutations may contribute. Regulatory failures may contribute. Cellular systems that normally maintain balance become disrupted. The immune system also depends heavily on regulated gene expression. Immune cells activate different genes when responding to infection. Inflammation involves coordinated changes across many signaling pathways. Even memory formation in the brain involves altered gene activity. Certain patterns of neural activity can trigger expression of genes associated with strengthening synaptic connections. The genome is not silent storage. It is continuously interacting with the living state of the organism. Scientists often compare gene regulation to an orchestra rather than a single instrument. Timing matters. Balance matters. Coordination matters. Different components must work together harmoniously. Another important idea is that many genes are regulated through feedback loops. A protein produced from one gene may later help reduce or increase activity of that same pathway. These loops help maintain stability inside cells. Biological systems constantly monitor themselves, adjusting, correcting, responding. Some gene activity follows predictable cycles. Hormonal rhythms fluctuate across the day. Sleep-related genes change activity across circadian cycles. Even metabolism shifts according to feeding and fasting states. Deep inside the body, molecular patterns are always changing quietly over time. The study of gene expression has transformed modern biology. Researchers can now examine which genes are active in different tissues under different conditions. This helps scientists investigate development, disease, aging, immunity, and brain function with much greater detail. Yet, despite all the complexity, the central principle remains surprisingly elegant. Cells contain the same DNA, but they use different parts of it at different times. This selective use of information is what allows trillions of cells to cooperate as one organized organism. And once a gene becomes active, the cell begins translating that stored information into physical molecular products. Inside the nucleus of the cell, DNA stores information carefully. Long sequences of molecular instructions written in chemical code. But DNA itself does not directly build tissues or carry out cellular work. The information must first be read, copied, and translated into functional molecules. This flow of information is one of the central ideas in molecular biology. Scientists often describe it with a simple sequence: DNA to RNA to protein. This process is sometimes called the central dogma of molecular biology. DNA stores the instructions. RNA carries and helps process those instructions. Proteins perform much of the actual work inside the cell. The process begins inside the nucleus. When a gene becomes active, the cell creates a temporary RNA copy of part of the DNA sequence. This step is called transcription. During transcription, the two strands of DNA separate slightly in a small region. An enzyme called RNA polymerase moves along one DNA strand. As it moves, it assembles a complementary RNA sequence. RNA resembles DNA in many ways. It also uses nucleotide bases. But there are important differences. RNA usually exists as a single strand rather than a double helix. And instead of the base thymine, RNA uses a related base called uracil. So, in RNA, adenine pairs with uracil rather than thymine. The newly created RNA molecule carries information copied from the gene. This RNA transcript acts as a working copy of the instructions. The original DNA remains protected inside the nucleus. In complex cells, like human cells, the initial RNA transcript often undergoes additional processing before use. Certain segments are removed. Others are joined together. Protective structures are added to the ends. This processed molecule is called messenger RNA or mRNA. Messenger RNA carries genetic instructions from the nucleus out into the cytoplasm of the cell. The cytoplasm is the fluid-filled interior where many cellular activities occur. Waiting in the cytoplasm are structures called ribosomes. Ribosomes are molecular machines responsible for building proteins. A ribosome attaches to the messenger RNA strand and begins reading its sequence. The information is interpreted 3 bases at a time. These 3 base units are called codons. Each codon corresponds to a particular amino acid, the building blocks of proteins. For example, one codon may signal the amino acid leucine. Another may signal glycine. Another may signal serine. The genetic code is highly organized and nearly universal across life on Earth. Many organisms use the same codon system to build proteins. This shared code reflects deep evolutionary relationships among living things. As the ribosome moves along the messenger RNA, amino acids are linked together one by one into a growing chain. This step is called translation. Translation converts nucleotide information into protein structure. A chemical language becomes a physical molecular product. Specialized RNA molecules called transfer RNA or tRNA help during this process. Each transfer RNA carries a particular amino acid. It matches its anticodon sequence to the corresponding codon on the messenger RNA. In this way, the correct amino acids are added in the proper order. The sequence matters enormously. Even small changes can alter the resulting protein structure and function. Once the amino acid chain is complete, it folds into a three-dimensional shape. This folding process is critical. Proteins function largely according to their shape. Some proteins form rigid structures. Others act as enzymes accelerating chemical reactions. Others transport molecules or send signals. The sequence encoded in DNA ultimately influences the final protein shape through this long chain of events. DNA to RNA to protein. Information flowing through multiple carefully coordinated steps. The cell performs these processes continuously. Right now, inside your body, countless ribosomes are assembling proteins. Thousands upon thousands every second. Some proteins are used almost immediately. Others remain stable for long periods. Some stay inside the cell. Others are exported outside. Insulin, for example, is a protein hormone released into the bloodstream to help regulate blood sugar. Hemoglobin, another protein, helps red blood cells transport oxygen. Collagen proteins provide structural support in skin and connective tissue. Antibodies help the immune system recognize threats. Ion channels help neurons transmit electrical signals. Proteins participate in nearly every aspect of life. And all of them begin through gene expression and molecular translation. The central dogma is sometimes presented simply, but real biology contains additional layers of complexity. Not all genes produce proteins directly. Some genes produce functional RNA molecules that help regulate cellular processes. Different RNA types play specialized roles. Ribosomal RNA helps form ribosomes themselves. Transfer RNA helps deliver amino acids. Small regulatory RNAs can influence gene expression. Cells contain intricate networks of RNA activity beyond messenger RNA alone. The process is also highly regulated. Cells do not produce all proteins constantly. Protein synthesis changes according to need. During exercise, muscle cells may increase production of certain proteins. During immune responses, defensive proteins become more active. During development, different proteins guide tissue formation. The body continuously adjusts protein production according to conditions. Accuracy is extremely important throughout these processes. Errors in transcription or translation can disrupt protein function. Cells therefore contain proofreading systems and quality control mechanisms. Misfolded proteins may be repaired or broken down. Damaged RNA may be degraded. Faulty components are often recycled. The cell is constantly maintaining internal order. Scientists have spent decades studying how ribosomes work in detail. These molecular machines are extraordinarily sophisticated. Yet they arise naturally through chemistry shaped by evolution. Life depends on these invisible, microscopic processes occurring continuously inside cells. Without transcription and translation, no proteins could be produced. Without proteins, cells could not function. Even the enzymes involved in transcription and translation are themselves proteins produced through the same system. Biology operates through interconnected cycles of information and chemistry. The central dogma also helps explain how mutations can affect organisms. A change in DNA sequence may alter RNA. Altered RNA may change the amino acid sequence of a protein. And altered proteins may function differently. Sometimes the effects are minor, sometimes significant. Much depends on where the change occurs and how it influences the final protein structure. Over evolutionary time, variation in DNA sequences help generate biological diversity across species. Small molecular changes accumulating gradually across generations. Yet, despite enormous diversity among organisms, the fundamental flow of information remains strikingly similar. DNA storing information, RNA carrying information, proteins carrying out cellular functions. Deep inside every living cell, these molecular processes continue quietly without awareness. Silent biochemical activity sustaining life moment by moment. And once proteins are assembled, they become the true working components that shape the structure and behavior of living organisms. Genes contain instructions. RNA carries those instructions through the cell. But proteins are the molecules that perform much of the actual work of life. Nearly every process inside your body depends on proteins in some way. Movement, digestion, memory, immunity, breathing, growth. All rely on proteins working continuously inside cells and tissues. Proteins are built from smaller units called amino acids. These amino acids are linked together into long chains during translation at the ribosome. Once assembled, the chains fold into complex three-dimensional shapes. Shape is extremely important. A protein's structure determines how it behaves, what molecules it interacts with, what reactions it can help perform, what physical roles it can carry out. Some proteins are flexible, others are rigid. Some act like tiny motors. Others function more like scaffolding or communication systems. One of the largest categories of proteins are enzymes. Enzymes help accelerate chemical reactions inside cells. Without them, many essential reactions would occur far too slowly to sustain life. Right now, enzymes inside your body are helping break down nutrients, produce energy, repair DNA, build cellular structures, regulate signaling pathways. A single cell may contain thousands of different enzymes operating simultaneously, quietly coordinating vast networks of chemistry every second. Digestive enzymes help process food molecules into smaller components the body can absorb. Other enzymes inside mitochondria help release energy from nutrients through cellular respiration. DNA polymerase helps copy DNA during cell division. RNA polymerase helps create RNA during transcription. Proteins are involved even in the processes that build more proteins. Structural proteins are another major category. These proteins help provide physical strength and organization throughout the body. Collagen is one of the most abundant proteins in humans. It helps support skin, tendons, ligaments, bones, and connective tissue. Strong fibers of collagen contribute to the body's structural integrity. Keratin is another structural protein. It helps form hair, nails, and the outer protective layers of skin. Inside muscles, proteins, called actin and myosin, interact to generate contraction. Tiny molecular movements repeated across enormous numbers of cells allow muscles to shorten and produce force. Even the beating of the heart depends on carefully coordinated protein interactions. Transport proteins help move substances through the body. Hemoglobin, the oxygen-carrying protein in red blood cells, binds oxygen in the lungs and releases it into tissues. This allows oxygen to reach cells throughout the body. Other transport proteins move nutrients across cell membranes, carry ions through channels, or help regulate fluid balance. Some proteins function as signaling molecules. Hormones, such as insulin, are proteins that help coordinate communication between tissues. Insulin helps regulate glucose levels in the bloodstream by signaling cells to absorb and store energy. Receptor proteins on cell surfaces detect chemical signals from the environment or neighboring cells. When molecules bind to these receptors, cells may change behavior in response. Immune proteins help defend the body against pathogens. Antibodies recognize specific foreign molecules and help target them for removal. Other immune proteins coordinate inflammation and cellular defense responses. The nervous system also depends heavily on proteins. Ion channel proteins regulate electrical activity in neurons. Neurotransmitter receptors help transmit signals between nerve cells. Proteins help maintain synapses, the communication points between neurons. Memory formation itself involves changes in protein activity and neural connections. Proteins are not permanent structures. Cells constantly produce new proteins and break down old ones. Damaged proteins may be recycled into amino acids and reused. This turnover helps maintain cellular function over time. The body is continuously renewing itself at the molecular level. Protein folding is one of the most delicate steps in biology. A protein must fold correctly to function properly. Some proteins require helper molecules called chaperone proteins that assist with proper folding. Misfolded proteins can sometimes accumulate and contribute to disease. Certain neurodegenerative disorders are associated with abnormal protein aggregation in the brain. Cells contain systems that monitor protein quality carefully. Faulty proteins may be repaired or targeted for breakdown. The balance between protein production, folding, and degradation is tightly regulated. Scientists estimate that humans may contain hundreds of thousands of different protein forms when considering variations and modifications. Even though the number of protein-coding genes is much smaller, This is partly because proteins can be modified after construction. Chemical groups may be attached. Segments may be removed. Interactions with other proteins may alter behavior. Protein networks are incredibly dynamic. Molecules constantly binding, separating, changing shape, and responding to conditions inside the cell. Temperature can influence proteins. Acidity can influence proteins. Salt concentration can influence proteins. The internal environment of the cell must remain relatively stable for proteins to function properly. This stability is called homeostasis. The body works continuously to maintain appropriate internal conditions. Evolution has shaped proteins over billions of years. Small genetic changes altering amino acid sequences occasionally produced new or improved functions. Useful variations were more likely to persist across generations. The diversity of proteins across living organisms reflects this long evolutionary history. Yet many proteins remain highly conserved across species. Core cellular proteins involved in energy production or DNA replication can show strong similarities between distant organisms. This reflects the shared ancestry of life on Earth. Proteins also contribute to visible traits. Pigment proteins influence coloration in skin, eyes, and hair. Structural proteins influence tissue shape and texture. Hormone-related proteins influence development and metabolism. In many ways, proteins are the physical expression of genetic information. Genes provide instructions, but proteins create much of the actual biological activity. Inside every cell, enormous numbers of proteins interact simultaneously in coordinated systems. Not as isolated molecules, but as parts of interconnected biochemical networks. The body depends on this coordination constantly. A heartbeat requires proteins. A thought requires proteins. Healing requires proteins. Sleep itself involves changing patterns of protein activity throughout the brain and body. And because proteins are built according to genetic instructions, small differences in DNA can sometimes lead to differences between individuals. No two humans are genetically identical, except in the case of identical twins who begin with nearly the same DNA sequence. Every person carries a unique combination of genetic variations inherited across generations. You may share certain facial features with your parents, similar eye shape, similar hair texture, similar height tendencies. Yet no one else has exactly the same combination of traits as you. This individuality begins at the level of DNA. Humans share the vast majority of their genetic code with one another. More than 99% of DNA sequences are typically the same across humans. But the small remaining percentage still represents millions of genetic differences spread throughout the genome. These variations contribute to biological diversity. Differences in appearance, differences in metabolism, differences in disease susceptibility, differences in how bodies respond to environments. Many genetic variations are extremely small, sometimes involving only a single base change in the DNA sequence. These single base differences are called single nucleotide polymorphisms, often shortened to SNPs. A single nucleotide change may have little effect, no effect at all, or occasionally a more noticeable biological influence. Most visible traits are influenced by many genes together, rather than a single gene acting alone. Eye color, for example, involves interactions among multiple genes related to pigment production and distribution. Height is even more complex. Hundreds or perhaps thousands of genetic variants contribute small effects related to growth and development. Nutrition and environment also play major roles. Human traits emerge through combinations of genetics and environment interacting continuously. When reproductive cells are formed, chromosomes are shuffled through a process called meiosis. During meiosis, chromosome pairs exchange segments of DNA in a process called recombination. This reshuffling creates new combinations of genetic variants. The sperm and egg cells produced are genetically unique, which means each child inherits a different combination of DNA. Even siblings with the same parents can differ substantially genetically. They inherit overlapping but distinct combinations of chromosome segments. Some inherited traits follow relatively simple inheritance patterns. Certain genetic disorders caused by mutations in single genes may be inherited in predictable ways. Scientists often describe these patterns as dominant, recessive, or linked to specific chromosomes. A dominant genetic variant can influence traits even when only one copy is inherited. A recessive variant generally requires two copies to strongly affect the trait. But most human characteristics are far more complex than simple dominant and recessive examples taught in basic genetics. Multiple genes interact together. Environmental factors interact with genes. Developmental conditions matter as well. Skin color provides one example of complex inheritance. Multiple genes influence pigment production and distribution. Sunlight exposure also affects skin pigmentation through biological responses in the body. Metabolism varies between individuals too. Some people process certain nutrients differently. Some metabolize medications at different rates. Some show differences in lactose digestion due to inherited genetic variations. Genetic diversity reflects both ancient human migrations and adaptation to different environments over long periods of time. Populations living in different regions faced different climates, food sources, pathogens, and environmental pressures. Over generations, certain genetic variants became more common in some populations due to natural selection or population history. Yet humans remain remarkably similar overall. The biological differences among individuals are small compared to the enormous amount of shared genetic information. Many variations have little noticeable effect. Some simply contribute to harmless diversity across the human population. Others may slightly increase or decrease risks for certain conditions without guaranteeing outcomes. Genetics often influences probabilities rather than certainties. A person may inherit increased risk for a disease, but never develop it. Another person with lower genetic risk may still experience it due to environment, aging, or other biological factors. Genes are important, but they do not function in isolation from life experience. The immune system illustrates this clearly. Certain immune-related genes influence how the body recognizes pathogens. But immune function also depends heavily on environmental exposure and physiological condition. The brain also develops through both genetics and experience. Genes help guide neural development and brain structure. But learning, memory, education, stress, sleep, and environment all shape neural connections throughout life. Scientists continue studying how genes influence behavior and cognition. These relationships are highly complex and rarely controlled by single genes alone. Many characteristics arise from interactions among enormous networks of genes and environmental influences combined together over time. Some genetic variants provide advantages in particular environments. Others may become disadvantages under different conditions. For example, certain inherited blood-related traits can influence susceptibility to diseases like malaria. Evolution often involves trade-offs rather than perfect solutions. Genetic variation is also essential for populations over evolutionary time. Diversity provides raw material upon which natural selection can act. Without variation, populations would struggle to adapt to changing environments. The process of inheritance connects modern humans to countless generations in the past. Your genome contains DNA passed down through parents, grandparents, great-grandparents, and many more generations beyond memory. Certain segments of DNA may have persisted across hundreds or thousands of years of ancestry. Quiet molecular continuity stretching through time. Scientists can study patterns of genetic variation to investigate human migration and ancestry. Shared DNA patterns among populations reveal relationships shaped by ancient movement and reproduction over time. Mitochondrial DNA, inherited primarily through the maternal line, has been especially useful for studying deep ancestry and population history. Y chromosomes can also help trace certain paternal inheritance patterns. Yet ancestry is always more interconnected and complex than simple categories suggest. Human populations have mixed and migrated repeatedly throughout history. Genetic testing technologies have advanced rapidly in recent decades. Scientists can now sequence individual genomes much more efficiently than in the past. Researchers study genetic variation to investigate disease risk, evolution, and population biology. Medicine increasingly uses genetic information to help understand certain inherited conditions and treatment responses. This field is sometimes called personalized or precision medicine. But genetics alone rarely provides complete predictions about human health or behavior. Biology remains deeply influenced by environment, development, chance, and complex interactions across many systems. Even identical twins, despite starting with nearly identical genomes, gradually accumulate differences over time. Mutations can occur in cells during life. Environmental experiences differ. Epigenetic patterns diverge. The genome provides a foundation, but life itself continuously shapes the organism. Still, every visible human difference ultimately traces back in part to molecular variation within DNA sequences inherited across generations. Tiny changes at the scale of molecules contributing to the diversity of billions of people. And sometimes, those changes in DNA arise not through inheritance alone, but through entirely new alterations appearing in genetic sequences themselves. DNA is remarkably stable. Cells contain sophisticated systems that copy, protect, and repair genetic information continuously. Most of the time, this stability allows organisms to grow and function reliably throughout life. But DNA is not completely unchanging. Sometimes the genetic sequence itself changes. These changes are called mutations. A mutation is simply an alteration in DNA sequence. It may involve a single base change, a deleted segment, an inserted sequence, or larger rearrangements involving entire chromosome regions. Mutations happen naturally. They are a normal part of biology. Every living organism accumulates some genetic changes over time. During DNA replication, occasional copying errors can occur. Most are corrected by proofreading and repair enzymes. But some changes escape repair and remain in the DNA sequence. External factors can also damage DNA. Ultraviolet radiation from sunlight can alter DNA structure in skin cells. Certain chemicals can damage genetic material. Some forms of ionizing radiation can break DNA strands. Even normal metabolism inside cells produces reactive molecules that may occasionally damage DNA. The genome exists in a chemically active environment constantly. Fortunately, cells contain elaborate DNA repair systems. Specialized proteins patrol the genome searching for damage. Some repair broken strands. Some remove damaged bases. Some correct mismatched sequences after replication. These repair mechanisms are essential for life. Without them, mutations would accumulate far more rapidly. Not all mutations have noticeable effects. Many occur in regions of DNA that do not strongly alter protein function. Some mutations change DNA sequence without changing the resulting amino acid in a protein. Because the genetic code contains redundancy, multiple codons can sometimes specify the same amino acid. Other mutations may alter protein structure significantly. A single amino acid change can occasionally affect how a protein folds or functions. Some mutations are harmful. Some are neutral. A few may occasionally provide advantages under certain conditions. Evolution depends on this variation. Without mutations introducing new genetic differences, populations could not adapt over long periods of time. Natural selection acts on inherited variation across generations. If a genetic change improves survival or reproduction in a particular environment, it may become more common over time. If a mutation strongly disrupts biological function, it may reduce survival or reproductive success and become less common. Most mutations are small, tiny molecular changes occurring silently inside cells. But across millions of years, accumulated mutations contributed to the enormous diversity of life on Earth. The wings of birds, the eyes of mammals, the nervous systems of complex animals, all shaped gradually through evolutionary processes acting on genetic variation. Evolution does not work toward predetermined goals. Mutations arise without planning. Environmental pressures then influence which variations persist across generations. Some mutations affect only individual body cells during life. These are called somatic mutations. They are generally not passed to offspring because they occur outside reproductive cells. Skin cells exposed to sunlight may accumulate somatic mutations over time. Many remain harmless. But if certain mutations disrupt growth control systems, cancer can sometimes develop. Cancer is fundamentally linked to changes in cellular regulation. Mutations in genes controlling cell division, DNA repair, or programmed cell death can contribute to uncontrolled growth. Yet cancer usually develops through accumulation of multiple genetic and regulatory changes rather than a single mutation alone. Other mutations occur in reproductive cells, sperm, or egg cells. These are called germline mutations. Germline mutations can be inherited by future generations. Every person carries some new mutations that were not present in their parents exactly as inherited. Genetic variation continues accumulating gradually across generations. Some inherited mutations are associated with genetic disorders. For example, mutations affecting hemoglobin structure can contribute to sickle cell disease. Other disorders involve mutations influencing enzyme function, nervous system development, or immune regulation. But many diseases involve combinations of genetic risk factors and environmental influences together. Biology is rarely controlled by single causes alone. Scientists study mutations using many different technologies. DNA sequencing allows researchers to compare genetic sequences and identify changes. Population genetics examines how variants spread through populations over time. Evolutionary biology uses genetic evidence to reconstruct relationships among organisms. Shared mutations can reveal common ancestry between species. Humans share substantial portions of their genome with many organisms. Shared genes and shared mutations reflect deep evolutionary history. Certain DNA sequences remain highly conserved across species because they perform essential functions. Other regions evolve more rapidly and accumulate variation more easily. Evolution operates continuously even now. Populations still experience mutations. Natural selection still acts. Genetic drift still influences variation. The process simply unfolds slowly across generations. Mutation rates themselves are relatively low per generation. This balance is important. Too much instability would threaten essential biological systems. Too little variation would limit adaptation over evolutionary time. DNA repair systems help maintain this balance carefully. Some organisms have evolved especially interesting mutation-related adaptations. Certain bacteria can exchange genetic material between cells. Viruses mutate rapidly because of high replication rates and reduced proofreading accuracy. RNA viruses in particular can evolve quickly under changing conditions. The study of mutations also transformed medicine. Scientists can identify some disease-associated variants through genetic testing. Cancer research increasingly examines mutation patterns inside tumors. Gene editing technologies such as CRISPR allow researchers to modify DNA sequences more precisely in laboratory settings. These technologies hold significant scientific and medical potential, though they also raise important ethical questions. Understanding mutations helps explain both disease and evolution. The same molecular processes that can disrupt cells also generate the diversity upon which evolution depends. Tiny alterations in DNA sequences, occurring silently across generations, helped shape the entire history of life on Earth. Yet DNA alone cannot fully explain what a person becomes. Because genes interact continuously with the environment surrounding the organism throughout life. Genes influence biology continuously throughout life. But genes do not operate alone. Every cell exists within an environment. And that environment constantly affects how the body develops and functions. From the moment life begins, cells respond to signals around them. Nutrients, temperature, hormones, chemical gradients, stress signals. The body is always interacting with its surroundings. Scientists sometimes describe this relationship as an interaction between genes and environment. Genes provide biological possibilities and tendencies. Environment influences how those possibilities unfold. A person may inherit genes associated with height potential. But nutrition during childhood strongly affects whether that potential is fully reached. Genes related to muscle development may exist. But physical activity influences how muscles actually grow and adapt. Even identical twins, who begin with nearly identical genomes, gradually become more different over time. Their experiences differ. Their environments differ. Their patterns of gene expression slowly diverge. The nervous system provides a powerful example of this interaction. Genes help guide early brain development. They influence formation of neurons, neural pathways, and signaling systems. But experience continuously shapes the brain afterward. Learning changes neural connections. Practice strengthens pathways. Stress influences hormone systems and brain activity. Sleep affects memory consolidation and cellular repair. The brain remains dynamic throughout life. Nutrition also interacts deeply with biology. Cells require amino acids to build proteins. Vitamins and minerals support enzyme function. Energy from food powers cellular activity. Poor nutrition can disrupt development and physiological balance. During early childhood especially, environmental conditions can strongly influence growth and health outcomes. The developing body is highly responsive to its surroundings. Exposure to toxins may also affect biological systems. Certain chemicals can damage DNA or interfere with normal cellular signaling. Air pollution, tobacco smoke, and excessive radiation exposure can increase risks for cellular damage and disease. The immune system is shaped by both genes and environment together. Genetic factors influence immune responses. But immune systems also learn from exposure over time. Contact with microbes helps train immune recognition systems throughout life. Physical activity changes the body as well. Exercise influences muscle cells, metabolism, circulation, hormone levels, and even gene expression patterns. During exercise, cells detect mechanical and chemical changes. certain genes become more active. New proteins are produced. Tissues gradually adapt to repeated demands. Bones respond to physical stress by remodeling themselves. Muscles can strengthen. Cardiovascular systems can adapt. The body is highly responsive to repeated environmental conditions. Sleep is another deeply important environmental influence. During sleep, the body performs many maintenance and regulatory processes. Hormone levels shift. Neural activity patterns change. Certain cellular repair systems become more active. Sleep deprivation affects cognition, metabolism, immune function, and emotional regulation. The brain and body require regular recovery cycles to maintain balance. Circadian rhythms help coordinate many of these biological processes. These rhythms are influenced strongly by light exposure. Specialized cells in the eyes help synchronize internal timing systems with the external day-night cycle. Light in the evening can influence hormone release and sleep timing. Darkness helps promote biological signals associated with rest. Stress also influences the body profoundly. Short-term stress responses can be useful in emergencies. The body releases hormones, such as adrenaline and cortisol, to prepare for action. Heart rate increases. Energy availability shifts. Attention becomes more focused. But chronic stress over long periods can strain many biological systems. Sleep may worsen. Immune regulation may become disrupted. Cardiovascular strain may increase. The body functions best when regulatory systems can regularly return to balance. Scientists study many of these interactions through epigenetics. Environmental conditions can sometimes influence chemical markers associated with DNA and chromatin structure. These changes may affect patterns of gene activity. Some epigenetic patterns are temporary. Others may persist longer. Researchers continue studying how environment shapes gene regulation across life. Importantly, environmental influence does not mean biology becomes infinitely flexible. Genes still matter greatly. The body operates within biological constraints shaped by inherited DNA. Instead, genes and environment function together continuously. Neither acts completely alone. Human behavior itself also emerges from this interaction. Genetics can influence tendencies related to temperament, sensation, learning, or risk for certain conditions. But culture, education, relationships, nutrition, sleep, stress, and life experiences also shape behavior profoundly. Biology is not destiny in a simple mechanical sense. Living systems are dynamic and responsive. Aging reflects this interaction too. Over time, cells accumulate damage. Protein systems change. DNA repair becomes less efficient. Stem cell activity shifts. Environmental exposures throughout life contribute to these processes alongside genetic influences. Some people inherit variants associated with increased longevity or reduced disease risk. But lifestyle and environmental conditions also influence aging outcomes significantly. Even microorganisms living inside the body contribute to the interaction between biology and environment. The human microbiome, communities of bacteria and other microbes living especially in the digestive system influences digestion, immune function, and metabolism. These microbial ecosystems interact constantly with diet and environment. Humans are not biologically isolated systems. We exist within larger ecological relationships. Modern medicine increasingly recognizes the importance of environmental context. Public health improvements, such as sanitation, nutrition, vaccines, clean water, and reduced pollution, have dramatically improved human lifespan and survival. These changes did not alter human DNA directly, but they changed the environments in which humans live. And because biology responds to environment, outcomes changed too. The relationship between genes and environment is not a simple competition between nature and nurture. They are deeply intertwined processes, influencing one another continuously. Every moment, your cells are responding to both inherited instructions and present conditions simultaneously. Molecular systems adjusting quietly beneath awareness. Yet, despite everything genes and environment can explain, there remains an even larger story hidden within DNA itself. A story stretching far beyond individual humans, back through the deep history of life on Earth. Every living organism on Earth uses DNA or closely related genetic systems to store biological information. Humans, trees, birds, fish, bacteria. Vastly different forms of life, all connected through shared molecular foundations. This shared biology points toward one of the most important ideas in science: evolution. Evolution describes how populations of organisms change gradually across generations over immense spans of time. Not through sudden transformation, but through accumulated variation, inheritance, and natural selection. DNA preserves a record of this history. Hidden inside genomes are traces of ancient relationships stretching back billions of years. Humans share a large portion of genes with many other organisms. We share genes with chimpanzees, with mice, with birds, even with plants and bacteria at deeper evolutionary levels. The percentages differ, but the underlying continuity remains remarkable. Core cellular systems are often deeply conserved across life. The genetic code itself is nearly universal. Across most organisms, the same codons correspond to the same amino acids during protein construction. This consistency strongly suggests common ancestry. Life appears to share ancient molecular origins. Scientists believe life began very early in Earth's history, likely more than 3.5 billion years ago. The exact origin remains uncertain and actively studied. But at some point, self-replicating chemical systems emerged. Over immense periods of time, these early systems became more complex. Natural selection favored molecules and organisms better able to survive and reproduce under particular conditions. Small genetic variations accumulated generation after generation. Populations diverged gradually. New species eventually emerged. Evolution operates without foresight or planning. Mutations arise naturally. Environmental pressures influence which variations become more common over time. Organisms better suited to particular environments tend to leave more descendants. Over long timescales, populations adapt. DNA allows scientists to reconstruct many of these relationships. By comparing genetic sequences across species, researchers can estimate evolutionary relatedness. Organisms with more similar DNA generally share more recent common ancestors. The evidence from genetics strongly supports evolutionary theory. Shared genes, shared mutations, shared molecular mechanisms, all pointing toward branching patterns of descent over time. Certain genes remain highly conserved because they perform essential functions. Proteins involved in DNA replication, cellular respiration, or ribosome function often show striking similarities across very distant organisms. These ancient molecular systems were so effective that evolution preserved them across billions of years. Other regions of DNA change more rapidly. These variations help scientists study more recent evolutionary changes and population history. Fossils provide one line of evidence for evolution. Comparative anatomy provides another. Genetics adds a powerful molecular layer connecting them together. For example, humans possess remnants of ancient genetic history within the genome. Some genes resemble versions found in other mammals. Some non-functional sequences appear related to older ancestral genes. The genome contains traces of evolutionary inheritance accumulated over deep time. Evolution also explains why organisms share similar developmental patterns. Many animals begin development using related genetic pathways controlling body organization. These shared developmental systems reflect common ancestry modified gradually across evolution. Natural selection is one important evolutionary mechanism, but not the only one. Random processes also influence populations. Genetic drift, for example, can cause certain variants to become more or less common simply through chance. Small populations are especially influenced by random genetic changes across generations. Migration also shapes evolution. When populations move and mix, genetic variants spread between groups. Human history contains countless migrations and interactions across continents over thousands of years. Isolation can contribute to divergence too. When populations remain separated for long periods, they may gradually accumulate enough differences to become distinct species. Evolutionary change usually unfolds slowly relative to a human lifetime. But across millions of years, small changes can produce extraordinary diversity. The feathers of birds evolved gradually from structures present in ancient dinosaurs. Whale ancestors once walked on land before adapting to aquatic life. Human ancestors evolved over long periods alongside changing environments and populations. The human species itself, Homo sapiens, emerged relatively recently in evolutionary history. Anatomically modern humans appeared roughly a few 100,000 years ago. Yet the molecules inside human cells connect us to far older biological systems. The enzymes copying your DNA today resemble ancient molecular machinery inherited across countless generations of life. Evolution does not produce perfect organisms. It works with existing structures and past constraints. Many biological systems reflect compromise rather than ideal engineering. The human spine, for example, evolved from ancestors with different body structures. Human eyes contain certain limitations shaped by evolutionary history. The recurrent laryngeal nerve in mammals takes an unusually long route through the body because of inherited developmental patterns. Evolution modifies existing systems incrementally rather than redesigning organisms from nothing. DNA mutations provide variation. Selection filters variation. Time accumulates change. Across billions of years, these simple principles generated the immense diversity of life on Earth. Evolutionary biology also helps explain disease. Some genetic variants that once provided advantages under past conditions may contribute to health problems in modern environments. Human biology evolved under very different lifestyles than many modern societies experience today. The study of evolution continues actively. Scientists use fossils, genetics, ecology, developmental biology, and molecular analysis together to investigate life's history. New discoveries constantly refine understanding of evolutionary relationships and ancient populations. Yet perhaps the most striking realization is how connected life truly is. Every organism alive today is linked through continuous chains of inheritance extending backward through deep time. No breaks, no sudden separations. Only branching continuity across generations. The DNA inside your cells carries molecular echoes of ancient organisms that lived long before humans existed. Ancient cells dividing. Ancient genes replicating. Ancient evolutionary experiments unfolding across oceans and continents over unimaginable spans of time. And now, after billions of years of inheritance, the same molecular system continues quietly inside your body at this very moment. Yet despite everything science has discovered about genes and DNA, many of the deepest questions about life and consciousness still remain open. For thousands of years, humans wondered how life worked. Why children resemble their parents, how the body develops, why illness appears, why living organisms differ from one another. Long before modern genetics, people observed inheritance without understanding the molecular mechanisms beneath it. Today, science has uncovered many of those mechanisms. DNA stores biological information. Genes guide protein production. Cells regulate gene activity continuously. Evolution shapes populations across time. We now understand far more about life than previous generations could have imagined. Yet despite all this knowledge, many mysteries remain. Biology is extraordinarily complex. Every discovery often reveals additional layers beneath it. Scientists continue studying how genes interact across vast networks inside cells. How consciousness emerges from neural systems. How aging unfolds at molecular levels. How life first began on early Earth. Even now, many answers remain incomplete. The human genome itself still contains regions scientists do not fully understand. Some DNA sequences have functions that remain uncertain. Some regulatory interactions are still being mapped. The brain presents even deeper complexity. Roughly 86 billion neurons connected through enormous networks. Proteins, electrical signals, and chemical interactions unfolding continuously. Consciousness emerges somehow from physical biology, yet no complete scientific explanation fully captures subjective experience itself. Memory remains deeply fascinating too. Experiences can alter neural pathways. Learning reshapes connections between neurons. Sleep helps stabilize certain forms of memory. The brain is not fixed. It changes continuously throughout life. Scientists also continue exploring aging. Cells accumulate damage over time. DNA repair systems become less efficient. Protein maintenance changes. Stem cell activity shifts. Yet aging is not caused by a single mechanism alone. It involves many interacting biological processes occurring together across decades. Research into genetics and medicine continues rapidly. Technologies for DNA sequencing have become dramatically faster and more affordable than in the past. Scientists can now analyze genomes at scales once unimaginable. Gene editing tools such as CRISPR allow researchers to modify DNA with increasing precision in laboratories. These technologies may help investigate inherited diseases and biological mechanisms more deeply. But powerful technologies also raise ethical questions. How should genetic information be used responsibly? What forms of gene editing should be permitted? How should privacy be protected? Science expands human capability, but societies must still decide how to apply that capability carefully. Modern genetics has also transformed medicine. Researchers can identify some inherited disease risks through genetic testing. Certain cancers are now studied partly through their mutation patterns. Some treatments increasingly aim to target specific molecular pathways. This growing field is sometimes called precision medicine. Medicine becoming more individualized according to biological differences among patients. Yet biology rarely offers simple certainty. Most traits and diseases involve combinations of many genes, environmental influences, developmental factors, and random chance together. Human life cannot be reduced entirely to DNA sequences alone. The environment matters continuously. Relationships matter. Nutrition matters. Sleep matters. Education matters. Culture matters. Biology and experience remain deeply intertwined throughout life. One of the most profound discoveries of genetics may simply be how connected life truly is. The same molecular language exists across organisms separated by immense evolutionary distance. The same 4 DNA bases repeating across nearly all known life. Humans often imagine themselves separate from nature. But genetically, we remain part of a much older biological story. The molecules inside your cells are built from atoms forged long before Earth existed. Carbon formed in ancient stars. Oxygen formed in stellar interiors. Iron produced in supernova explosions. Over billions of years, those atoms became oceans, rocks, chemistry, cells, organisms, ecosystems, and eventually conscious beings capable of studying their own origins. DNA itself has no awareness. It is chemistry, molecular structure, information storage shaped by evolution. Yet, through countless generations of life, that chemistry contributed to nervous systems capable of memory, emotion, imagination, and reflection. Right now, while you rest quietly and listen, trillions of cells inside your body continue their activity. DNA replicating, proteins folding, neurons signaling, mitochondria producing energy. Silent molecular processes sustaining life moment by moment. Most of this happens completely outside awareness. Your heart beats without conscious control. Cells repair damage automatically. Genes activate and deactivate continuously. The body is an enormous coordinated system built from microscopic interactions. And despite all modern technology, living organisms still possess astonishing complexity. A single human cell contains molecular machinery far beyond anything humans have engineered independently. The more science learns, the more intricate biology often appears. Perhaps that is one of the quietest lessons hidden inside DNA. Life is both structured and dynamic, ordered and changing, stable and adaptable at the same time. 4 simple molecular bases repeated across billions of positions, giving rise to cells, tissues, ecosystems, and conscious minds capable of wondering about themselves. And tonight, somewhere inside your cells, ancient genetic patterns continue the same process they have carried across unbroken generations for billions of years. Quietly, patiently, continuously. As your body rests, the molecular story of life keeps unfolding softly in the darkness.